All right, good afternoon or good morning, everyone, depending on where you are joining us from today. We are so excited to have everyone here for our virtual field trip, Exploring the Pacific with Schmidt Ocean Institute. We have a great virtual field trip lined up for everyone today. But before we get started, just a little bit of housekeeping. My name is Allie Toomey, and I am the Education Coordinator with EarthEcho International. We are so excited to have classrooms from all across the country and the world joining us today as we explore the research vessel Falkor and the tropical waters of the Pacific. This hangout thrives on participation from all of you. We love to hear your questions. There are two ways for you to ask questions today depending on how you're watching. So if you are watching on our website, underneath of that YouTube viewing box, there's a Google question form. You can submit as many questions as you want through there. If you're watching it on full screen, you may have to minimize to be able to see that question form, but you're able to submit as many questions as you'd like through there. If you're watching through Google+, the Q&A app is active today. You can submit questions through the Q&A app right on the side of your viewing screen. If you see someone else that has the same question that you have, go ahead and give that question a +1. That way we know that there are lots of people with that same question, and we'll try our best to get all of those answered for you today. You can start asking questions right away. No need to wait until the end, but we may not get to them right away, but we will be sure to answer as many as possible. So now that we're ready to get started, I'm going to introduce everyone that we have with us today. First, I want to introduce the two classrooms that we have on camera. From Honolulu, Hawaii, we have Miss Yagi's science class. She has about, I think, 2 classrooms full of students with her today, so they are really excited. They are quite close to where the research vessel is, and a little bit further away in Coventry, Ohio, we have Mr. Dudley's class. They are with us today as well, so we're really excited to have them, and they have some questions about how the issues going on in the Pacific affect people in all different places of the country, even those that seem pretty far away. And so while we get started, I am going to introduce Dr. Mac Saito. He is the chief scientist on this cruise, and when he's not cruising around on a research vessel, he is an associate scientist at the Woods Hole Oceanographic Institute. So go ahead and take it away. Oh, and let's see, you guys are muted, so you'll have to unmute on your screen.
There you go. All right. Good morning. Can you hear me okay? So, good morning. We are on the RV Falkor in the center of the Pacific Ocean, several hundred miles south and east of Hawaii. And we are out here. We have about 10 science party from Woods Hole, Southern California, and Maryland, as well as 2 one artist and a videographer, as well as a undergraduate from University of Hawaii. And then the crew is about 23 on this ship. Just going to tell you a little bit about what it's like, life is like here, out here, and then I'll transition into talking about some of the science that we're doing out here, because I know a lot of people are curious what it's like to be out at sea. The ship it's a big ship. It's about 270 feet long, and we've been going through some sea states. We have 15 to 20 mile an hour winds out there, and some swell as well. So you might see us bobbing around like this. It's because we're experiencing some waves. But in general, the ship rides really nicely, and we're getting a lot of great science done out here. Just want to check before I go on. You can hear me okay?
Yes,
yes,
we can
no
hear
nods?
Yes, yes.
Great, thank you. You know, life. We're basically living out here for a month away from home on the ship. The distances in the oceans are so far that we can't go back to shore easily, and we have a rough speed of about 12 miles per hour out here. So we're basically out at sea land for that entire time. Maybe we'll see some islands later on. And so that really changes the way we do science compared to people back in laboratories. We have to bring everything we need with us out here. So we brought a lot of stuff. I joke we pack heavy, We have a big container worth of gear that we brought because we need not only the gear we have, but the backups in case things break. And the ship has a lot of food, a lot of frozen food, lasts the whole time, a lot of fuel. Water, we all sleep in cabins with beds or bunk beds, usually 2 or 3 or 4 per room. The ship has a full galley. Each cabin has its own bathrooms, or shared bathrooms and showers and things like that. And it's a big ship. There's a laboratory down the hallway and a dry lab, so where we have all of our gear, and also there's living spaces like this one. This is a library that we're in, and a galley downstairs, and the bridge upstairs, and a lot of deck space on the back of the ship where we deploy our gear. And that's that's actually something that's pretty specific to oceanography, is that there's a lot of heavy machinery involved in getting samples out of the oceans, because we're over typically 4 to 5 kilometers, 4 to 5,000 meters of water, and we're trying to sample all that water. And so in order to do so, you can't just, a bucket and rope doesn't work very well unless you're just trying to sample the surface waters. And that's what people did historically for hundreds of years. They would sail around in sailing ships and just drop buckets over and write down the temperature and things like that. But these days we're trying to study the entire ocean depths, and that means having big spools of wire and synthetic wire and big winches and having to wear hard hats and working on the fantail with steel-toed boots to get these samples from thousands of meters down below and bringing them back up, and having gear that can handle the weather states of being at sea, and knowing how to work under those conditions, high sea state conditions, as well as having gear that can withstand the pressures and the roughness of the ocean state. The oceans can be really rough on equipment. I don't know, how many of you have been out swimming and surfing and felt how strong the waves can be, and you can imagine what that's like when gear is out there for long periods of time. And that's a little bit about life on the ship. One thing that I think people are curious about is what communications are like back home. When I first started going to sea in the mid '90s, we didn't have any internet. We had a phone you could use that was like $20 a minute, and to a satellite link up. So if you really had an emergency and you wanted to call home, you could do that. But pretty much it was no communication with home that whole time. And now this is amazing that we can actually do this internet communication with you guys in real time back there. Let's hope that the bandwidth stays. The ship has 2 satellite transmitters in order to enable this, 2 big domes on the top of the ship that are communicating with satellites.
One question.
Should I
from teachers in different classrooms is, how long do you stay out, and how many cruises do you go on per year?
Yeah, so this cruise is, I think we're 28 days. Cruise length, or the expedition lengths, we'd like to use the word expedition because people imagine we're on big cruise ships otherwise, and it's really a lot of work out here. It's not playing shuffleboard or something like that. We work long hours, tend to work 12 plus hours a day, or work until we just are too tired to work anymore. But yeah, for myself personally, I tend to go out about a month a year, and then that gives me enough data to work up for the rest of the year. The ship time itself is a really valuable resource, so getting access to the sea, getting out here, it's an expensive operation to run. So you really prepare a lot to get ready to go out to sea, and then you try and plan all your experiments and pack all your sample and gear and your lab equipment. And when you go out there, you just try and work really hard because it's such a great opportunity to be out here. Good.
Great. Awesome.
Any other thing?
Awesome. I think that people are excited to hear a little bit about the research that you're conducting and how exactly you guys are actually taking samples while you're on the ship.
Sure, that sounds great. Should we switch over to the slides that I have?
Yep, I'll do that.
So yeah,
I'll do that for you.
Great. So yeah, out here we are focusing on the low oxygen regions of the oceans, and these are regions that have oxygen that can get down as low as a few micromoles per kilogram, and that's quite a bit lower than what we see in the surface waters that are in equilibration with the atmosphere and where all the phytoplankton are. This first slide just goes to show some of the complexity of the oceans. Carly, it's actually an animation. if you want to push, I don't know if it'll come through, but there's a little button on the bottom left. If you mouse over, yeah, a little farther. This is some model output from a colleague of mine at MIT that they're using computers to predict where the algae in the oceans are and what major groups of algae are out there. And you can see all the different colors that are part of it. The greenish colors are some of the smallest algae, what's known as the cyanobacteria, and whereas the yellow and the red are larger algae, some of the diatoms and other species that tend to live in cooler waters and more productive waters near the coastlines. And so they're an example of the complexity of the biological systems that are out there. And just like we think a lot about the vegetation on land, this is really the vegetation of the ocean, or component that's doing the photosynthesis. So this is sort of the base of the food web out in the oceans. But we're also really interested in what's happening underneath it, and that can be far below where the light is. The light only penetrates about 150 to 200 meters deep out here. It's much shallower in lakes and in coastal waters because there's so much more biology in the water, so many more microbes in the water that they self-shield the light. But out here in the open ocean, it's more of a desert. There's less nutrients, and so light penetrates very deep. That's what, almost 600 feet deep. Okay, let's go to the next slide. And so today I'll talk about some of our objectives, and really we're talking about several intersecting things here. One is studying the microbes that are out here, and by microbe I mean organisms that are just very small. Taxonomically, that includes bacteria. It also includes the archaea. It also includes algae, eukaryotic phytoplankton. So really, from the three major domains of life, but because they're all small and free-floating and contributing to the ecosystem, we generally use the more common term microbes to describe them. And then we're also talking about, we're also interested in how nutrients and micronutrients influence those microbes. And when I talk about micronutrients, I'm talking about primarily metals. You know how we need iron and zinc. You know, you can take zinc pills when you've got a cold or something like, or zinc lozenges, and people take vitamins like vitamin B12. Those are all metal-containing micronutrients, and basically all life must have them to survive. And so there's relatively little known about micronutrients in general compared to major nutrients, which are nitrogen and phosphorus and carbon, and in some cases silica. But together, all life basically needs to accumulate these nutrients to make the biomass that we are constituted from microbes to humans to really everything. And in the oceans, that's actually a really important issue. Acquiring those nutrients can be very difficult because they get used up, and then especially in the upper water column where light is penetrating, they get used up. And so that tends to drive a lot of the ecosystems out here, this access to nutrients. Just like we need food to eat, all life needs to have nutrients to build itself. And then the third component we're really studying out here is how those two phenomena then intersect with the low oxygen waters of the Eastern Pacific Ocean. And the Eastern Pacific has a large low oxygen region that extends all the way from the what we call the eastern boundaries near South America, all the way out past Hawaii in the northern hemisphere, and a little bit farther out in the southern hemisphere. And I'll talk about them a little bit more later, but they're really fascinating regions. They're essentially reactors for the oceans, and they're a little bit different than the coastal dead zones that we hear a lot about in the news, and that I know that I hear there's a Lake Erie, Northern Ohio classroom connected here as well. So it's similar to that in how they're formed, but it's not quite as extreme to the point where we have fish kills and things like that. And these are largely natural phenomena, the ones that we're studying out here, but we believe that they're being impacted by humans and becoming stronger perhaps because of climate change activities. And so because they're such important reactors to the ocean, that's one reason we're out here to study. Oh, here we go. There was a question. It said, it disappeared though.
Why are there no oxygen zones?
Oh yeah, why? Yeah.
They move around the ocean with circulation.
That's right. So why are they there? So they're there for a couple reasons, we think. One is that the ocean gyres tend to circulate in the northern and southern hemispheres, and so in the Pacific Ocean you have the northern and southern gyres that circulate, and so the corners to the east are sort of out of that circle area of where the gyres are circulating, and so they tend to have a little less circulation, and so they're not getting what we call ventilated as easily, and so the oxygenated water of the atmosphere and the upper water column don't get to mix as easily down below. Also, because of the way the circulation works, that water in the eastern boundary is being pulled up from depth, so deep water is coming up. Again, that's preventing those waters from equilibrating with the atmosphere. And then thirdly, there's a lot of productivity. Those microbes that I was talking about, there's a lot of nutrients that come up when water is upwelled, and so that creates a lot of productivity. And then that growth of the microbes fuels the ecosystem, and then that rains down matter and dead organic matter as they die off and are eaten by zooplankton and larger components of the food web. And as that sinks down, that organic matter then is remineralized, and as it does so, its oxygen is used to degrade it, just like we breathe oxygen to degrade organic matter, sugars and food and stuff like that. That's the most efficient reaction for life as well, and so that depletes the oxygen in those regions as well. So that combination of slower circulation in the shadow zones and enhanced productivity causes these large oxygen minimum zones then can persist there for a while because of that poor circulation.
And Mac, we had
so
a
these
question.
Are three.
Oh, sorry, Mac. We had a question that came in from a school in Seattle, and when you talk about how large these oxygen minimum zones and oxygen deficient zones are, how large are we talking, and what's like the largest one that you've ever seen?
Yeah, you know, I'll show a slide a little later about that, but they're really large. They're, you know, here we are south of Hawaii, and there's an oxygen minimum zone extending from here all the way back to the Costa Rica, Mexico, and areas. So that's basically, it's going halfway across the Pacific Ocean. So they can be tremendously large, and you know, they're a relatively modern feature of the modern oceans. You know, before photosynthesis was everywhere, before photosynthesis evolved, the entire oceans were probably like this. So it's interesting to imagine, you know, billions of years ago how things would have been different. So yeah, so one of our major emphases on this cruise is how do we try and study these interrelated processes, and one of the ways we do that is to use protein measurements. And the reason for this is that the microbes are very small, so we want to see what they are doing and who they are, who's there, and what they're doing. And one really nice way to do that is some of these new methods that are coming out in biological and biochemical research involving the omics type studies. And that's this sort of revolution in molecular biology and biochemistry, where we can start going into organisms and seeing what their genes, their DNA, and their RNA, and now most recently their proteins are. And that's one thing we've really been specializing in, is trying to measure these proteins directly. And one reason for that is that one reason why we're excited about this is that proteins tell you what an organism is doing and what it's really expressing and feeling at that moment in time to anthropomorphize it. And we're also complementing that with a bunch of chemical measurements as well to see what's happening to the ocean chemistry as well. So I'll introduce these as I go along. Next slide. So to back up a little bit more, I talked about these microbes, and we really think that there are the engines that drive the planet's chemistry and life, and allow life to to exist on Earth. And that's a colleague had this nice analogy that it's the microbial engines that drive the planet. And I wanted to introduce this word biogeochemistry or biogeochemical cycles. And it sounds like a big, complicated word, but it shouldn't be. It's basically the study of the biology and the chemistry and the geology, as well as the physical processes that govern the composition and habitability of the environment. Another way to describe it is elemental cycling. And at essence, you know, in the classrooms and in textbooks, we tend to want to study each of those disciplines separately. But in ocean sciences, here we are floating over 5 kilometers of oceans, and all of those processes are happening together and are inextricably tied together. So we have no choice but to consider them together, and that's really what biogeochemistry is trying to get at. And it's really reflecting all the different reactions that life can do. And in particular, microbes are very good at conducting chemical reactions, and that's what this figure here shows. A whole range from oxygen photosynthesis, oxygenic photosynthesis, which is basically photosynthesis as we know it, to what came before that, anoxygenic photosynthesis, as well as different ways to breathe and gain energy from consuming organic matter, whether or not you use oxygen on the right in that gray box, or other molecules such as nitrate or manganese metals or sulfur molecules. Everything has got to make a living, making energy and making biomass on the planet in order to sustain their biomass and to perpetuate life. And microbes have forever been the key and the foundation for that, and allow the system to Earth to be habitable. And this has been going on since the beginning of life on Earth. And so that's one reason, sort of really stepping back deep in time. It's really fun to think about how the planet evolved with the chemistry of the planet, and how those two things were really inextricably tied together, and what that means for today. And these oxygen minimum zones provide a really great clue to that, because they are very similar to what ancient oceans were like and the ancient planet was like. Plus, they are really key contributors to the Earth today. We'll do a next slide.
Mac, before we get to the next slide, I think actually Miss Yagi's classroom had a question about education. Do you guys want to ask that, or do you want me to ask that for you? Go ahead.
You can do it.
Okay. Well, they were just wondering, as we're talking about all of these different intersecting areas of science, what is the general amount of, or what type of educational background do you need to work on a research vessel like the Falkor?
That's a good question, and there's probably not one answer. Because there's the science party that's on here, and a lot of us have science major backgrounds. Mine was biology and chemistry and environmental studies from my undergrad days. But in our science party, we have a lot of biologists and chemists. But really, any of the sciences for the science party, geology, chemistry, biology, physics. We recognize that there's not that many undergraduate oceanography programs. So in our graduate program, we look for students. We don't expect people to come in with oceanography majors. If they have strong foundations in science, that's what's important. And then the ship has a lot of engineers and technical people, as well as the captain and the mates, who have a lot of navigational experience and sea-going experience, as well as other technical capabilities. Yeah, like Leighton over here.
Yeah, my background's background's from computing, so my actual degree's in computer science. So I look after all the scientific hardware on board the multi-use system. So mine isn't actually a science background, and a lot of people who actually support the science don't come through a science channel. So as we're saying, there's many routes that you can take to actually get onto a research vessel like Falkor.
Great.
Great, thank you.
So, yeah, sure. I'll continue on. So, just to continue on how the oceans are influencing the chemistry of the planet, one of the main ways that they do this, and that is of broad interest, is that they contribute to the oxygen and the carbon cycling of the planet. The oceans are 70% of the Earth's surface, and they have what's known as the biological pump, which is the process of photosynthesis in the upper ocean, where plants or the algae really are growing, and in doing so they use carbon dioxide, just like trees and plants do on land. But that causes CO2 to then go into the ocean from the atmosphere to replenish the dissolved carbon that's there. And then eventually some of that phytoplankton is eaten, and the parts that are eaten either go up into higher trophic levels into fish, and some fraction of it also sinks into the deeper ocean. And that fraction that sinks into the deeper ocean is what we call the biological pump. Some end up an even smaller fraction, maybe 1% of that, will get down to the ocean floor. And that's because we're over such a tall water column of water that having something sink all the way to the bottom before it's completely digested back into CO2 is hard to do. But still, when something sinks below the upper few hundred meters, it's effectively removed from the atmosphere for decades to centuries. And so that's the ocean, one of the ocean wave mechanisms of removing carbon dioxide from the atmosphere. The other mechanism is simply what's known as the solubility pump, and that's on the right here, where that as the oceans circulate, that what we call ventilated water then goes into the subsurface, and then it's effectively removed from the atmosphere as well. So those two processes, the biological and the solubility pump, are the key mechanisms for removing carbon dioxide from the atmosphere, and they're both actively reducing the amount of CO2 that humans are putting up in the atmosphere. Without those two mechanisms, there would be a lot more CO2 in the atmosphere, and global warming would be a lot worse than it already is. So one of the big questions we have is how are carbon and oxygen, as well as all the other elements that we know are biologically relevant, cycling in the oceans, because they are important to understand the ocean's influence on the carbon cycle of the planet and how the planet may respond to climate change as a result. Next slide. And so out here we're studying really some of the very smallest microbes to some of the larger ones, and really the very smallest microbes that have been relatively recently discovered. In the C panel in the upper right, that's an archaea that was really only recognized, maybe a decade and a half ago, as being really fundamental to the oceans, as well as being extremely abundant. It's really one of the most abundant microbes in the oceans, and really it shows how young the science is. Just 20 years ago, we really didn't recognize how abundant it was. Part of that reason is that it's actually very, very hard to grow. If you in a lot of laboratories, you'll take you take bacteria and you put it on a plate that's got rich in nutrients, and you get a lot of things that grow up. That's sort of the traditional microbiological way of cultivating microbes. For these microbes that live in these very low nutrient environments, they don't typically respond to that. That's actually too much nutrients all at once. So we get other things that grow up that are rarer, and the more abundant things won't grow up. And so these microbes had remained hidden for a long time. To show how small it is, that little bar there is a 0.1 micron scale bar. So these microbes are only a few tenths of a micron in size. And the other two panels you can see, that's a bacterium, and all the little layers and bumps there are compilations of enzymes that are doing a bunch of reactions. We had a question of what kinds of tools are we using, and we're using a variety of tools, from sensors on the ship. We have a lot of sensors that are detecting different parameters that can measure oxygen and temperature directly, as well as a lot of water samples we're bringing up to measure the chemistry as well. Okay, next slide. And then we're also studying some of the larger microbes out here. This is one that's called ocean sawdust from old-time mariners, because they would see this thing that looked like sawdust floating across the surface of the ocean. And it's a cyanobacteria that makes these nice tufts and puffs, as they're called. It's usually a few millimeters in size when you see them in these colonies, but they do not only photosynthesis, but they also can take nitrogen out of the atmosphere and then provide nitrogen to the oceans. And nitrogen is interesting because it's sort of in the reverse of carbon. The atmosphere has actually relatively little carbon relative to the oceans and the land masses, but for nitrogen, most of the nitrogen is up in the atmosphere, and there's less in surface oceans. And so the process of bringing it into the oceans from the atmosphere is very important, and these microbes are the main way of doing so. And so my colleague Eric is out here studying those as well. Okay, next slide. And one of the reasons we want to study these organisms is because we know that the Earth is changing, and the organisms are trying to adapt to that, and we want to know how. And so recently, some colleagues found that microbes are evolving in response to higher CO2 levels, and they're evolving in a way that is irreversible. They sort of get stuck in a high CO2 mode. When you drop CO2 back down, they don't adjust back, and so that was kind of a surprise to us. That, the ecosystems are adapting, but then they're not adapting back. What does that mean in the future? Or do we just not give them enough time to adapt back? There's those are complicated questions, and to find those answers, we have to do a lot of studies to figure out what organisms will do. So this is just an example of how one organism is responding, but there are many organisms out here that we need to try and figure out what they're doing. Next slide. And so we're also studying some of the bigger organisms too. Diatoms are these beautiful, larger eukaryotic algae that form these silica glass houses. Their outside is actually made of silica, which is a glass material, and they have different shapes. You can see here they can be round pennate diatoms, as they're called, and longer chain-forming diatoms. But they're beautiful, and they're really important in carbon fixation, especially in coastal areas and in the polar regions. Next slide. And so here's the picture of where the oxygen minimum zones are out here. The purple areas are the low oxygen regions of the oceans. And there are effectively 4 or 5 of them, major ones in the oceans you can see labeled by in these maps. we're in this one in the Pacific Ocean, and you can see that tongue along 10 North that extends well across the basin. That's the one we are studying and going east through. We're going to go to about 140 west and then start heading south. But these are not at the surface. They're down several hundred meters, 200 to 500, 600 meters, depending on which one. But researchers have compiled data over the past few decades. You can see that on the right panels, D&E going back to 1960. And these regions look like they're expanding. You can see that from 1960, they are going, that's pressure, which is similar to depth. So they're going from roughly 200 to 600 meters. But as you move forward to 2010, those regions are expanding and going closer to the surface and going deeper in the water column in both the D and E boxes, which correspond to the Pacific areas that we are over. And so that's the interest in trying to understand what's happening to oxygen in the oceans and trying to study what it means if the oceans are gradually deoxygenating. And many of the microbes that we're interested in thrive in these low oxygen areas, and they're doing important chemistry too, influencing the carbon and nitrogen cycles. So we want to understand what reactions are happening in there and then how this deoxygenation may affect it. All right, next slide.
Mac, before we head on to the next slide, some of Mr. Dudley's students from Coventry had a few questions. Do you guys want to jump into some of your questions?
Sure, we're down to 2. School just ended here in Akron, Ohio, so it's 30. So here have a question though. How do we know for sure that nutrients in oxygen mushrooms?
Sorry, it's hard to hear you. Can you say it or get close to the microphone?
Yeah. What's up, Josh? How do we know for sure that activity changing in oxygen levels?
Yeah, how do we know for sure that the oxygen is changing and the activity is changing? Yeah, that's a really great question, and that's, scientists, we're a skeptical bunch, and that is something that we want to know is really happening. And so, so researchers compiled these data sets. If you look carefully on the panels on the right, all these little white dots that are going up and down, those are data sets that have been compiled to make that that data plot. And so that's one of the first questions. How much data do you have, and is it enough to say this phenomenon is really happening? And then, how good is the data? What is the error associated with those measurements, and do we believe it? And those are really important things that scientists really spend a lot of time thinking about, trying to make sure what we think is happening is really happening. Are the phenomena real? And I think it is a little early to know the extent of change and how important deoxygenation is going to be. But I think this is an observed data set, and I think it is at least presents some compelling questions and raises awareness of saying, hey, this looks like something that we should be focusing on being out here studying. But I think that, the answer is really better measurements and more measurements, and that means getting ships out here to make those measurements and being out here like we are. But the oxygen sensors tend to be pretty good. Right before this cruise, the ship, we've got 3 oxygen sensors on 2 different samplers that we have out here, and all 3 of them were calibrated just before this cruise to make sure we get really robust measurements. Good. Did you have another question in Ohio? No? Okay. I should say I have a special affinity for Northern Ohio. I used to work for the Lake Erie division of the Ohio Geological Survey, so I'm excited to see you guys on here. Okay, next slide. So one of the ways we're trying to understand this is to get understand the interplay between the microbes and the low oxygen, is to measure the proteins of the microbes that are out here. And proteins are exciting because they're roughly half of the weight of the biomass in an organism. And they have these beautiful 3-dimensional structures, as you see here. And as molecules, they're really the molecules that are doing a lot of the work that life needs done. They do a lot of the reactions, be it the nitrogen fixation reaction I talked about before. That's this protein on the left. The red dots in it are where the iron is that conduct the chemistry, and then all the twirls and things are the amino acid chains. And so that ocean sawdust organism I talked about before is full of this enzyme. And then the one on the right is a cytochrome, and it's got an iron in it as well. That red dot inside a purple heme molecule, and then the rest of it, the ribbons are the protein amino acid chains. and that's doing a lot of the electron transport in photosynthesis and in respiratory organisms as well. And so these are the key enzymes that are doing reactions out here, or these are just two examples of many of them. But as you can imagine, if you live in a place that has very little metal or very little nitrogen, those are both key components of proteins. And so how do you make a living doing that? And that's a real challenge. And so that's one thing we're out here doing. By measuring the proteins of organisms, we can see what they're doing. Next slide. And so one of the ways we're doing this is we're using a new medical technique called proteomics, which is the measurement of many proteins simultaneously. And so that involves taking a bunch of biomass that we're collecting on filters. That so we're either bringing water up from deep in bottles like this sampling bottle. Here we go. Here, this is an X-Niskin sampling bottle. That is it full of water? Should be good. The tops come off. It's spring-loaded. It goes down open. I don't have to bang the ship up too much. And we can drop this to whatever depth we want, down to 5,000 meters. It attaches to the wire, or goes onto what we call a rosette with these clamps and these buttons. And then when we get to a pre-programmed depth, it'll close up, and then it'll bring this water. I think it's about 12 liters. Bring it up to the surface, and then this goes on a rosette where we have 24 of these. And we can drain them and pull the water out, and then study what's in it, the microbes or the water chemistry. And then we take those samples and we'll pull the micro biomass out of them and break them up into smaller pieces of proteins, into peptides, and then inject them into a mass spectrometer, which is a machine that basically measures weights, but it measures weights of really small molecules and measures them really, really fast. It can collect hundreds of thousands of weights over several hours. And then we use genomic data information, the DNA information, to try and understand what proteins were there, because proteins are made from DNA and via RNA. And so this is a technique we use back in the laboratory to do that. Next slide.
Before we head on to the next slide, we had a student with a question as we were talking about these really, really tiny organisms and proteins. How are these affecting larger animals within the ecosystem at the same time? What are some of those effects that you're having?
Right. So these are the base of the food chain. So without the microbes, we don't get to see. We don't get the zooplankton that eat on them, and the fish that eat on them, and the mammals that eat on them. So really, the extent of their growth really controls much of the whole ecosystem. And there's also other attributes. Some of these algae can make toxic molecules, and that can affect the fish as well. So they're really the base of the food web in that sense. And so the techniques that we're developing that we're using out here are really being pulled from medical research. And one of the reasons for that is the medical community is much bigger than the ocean science community, probably a factor of 100 or 1,000 times bigger. And so there's a lot of money and effort going into building good technology there. But fortunately, life is life, so the same techniques that work on humans will also work on the oceans. And so we've been adapting these methods for that. And so we call them metaproteomics. And there's also something called metagenomics, and that's meant to be the study of proteins or genes in a wide community. We're not just measuring one person or one animal. We're measuring many organisms mixed together out here, because we can't physically separate them because they're all mixed together in the oceans. At least we can't easily do it. Next slide.
Very interesting.
It's been a lot of fun, actually, trying to connect and bring these new methods in. And so much of science is really driven by technology. Like I said, 200 years ago, people would go out with buckets and thermometers. Now we can go out and use mass spectrometers to really get a pulse of what the ocean is doing. And so this is our cruise track. We're going from Hawaii. We've gone straight south and done a few stations, and now we're on that diagonal notch that's going northeast, and we'll go east right across the oxygen minimum zone, and then when we get to 140, we'll go straight south across the oxygen minimum zone, and then across the equator, which is low in iron, and then duck into Tahiti. All right, next slide. So that's about 4,000 kilometers of ocean. That's a long way, and it's going to take us. I think we have about 17 or 18 days of just transiting at 12 knots to do that, and then we stop. We'll be stopping for 12 or 13 days, broken up into either 2 to 24-hour increments to do the sampling, where the ship comes to a halt and we put the wires over and with these bottles and things to get our samples. So that's a big part of being out here, is what we call wire time. How do we budget our time sampling from the oceans, because the time is so valuable, and we can only have one wire down at a time, because if you imagine if you have 2 wires out at 3 kilometers and the ship turns, they would get tangled. And so we can't do more than one sampling operation at a time. And one of our goals is really to try and understand what the whole of the oceans are doing. And because the oceans are so huge, that's really difficult. This is an example of what we've been inspired by, something called the Geotraces program. I encourage you, if you're interested, to go to this website, egeotraces.org, where they have a lot of pictures like this. This is a very new program to measure micronutrients in the oceans. Before 1985, there weren't any good measurements of metals in the oceans, or at least not of iron in the oceans, because we're on a big metal ship, and it's very easy to contaminate for metals when they're in incredibly low levels like they are in the oceans. And you can see the red hot spot down in the bottom of the Atlantic Ocean. That's a hydrothermal vent contributing iron in over several hundreds or thousands of kilometers. And then up in the upper water column, you can see how it's purple. Those are really low iron concentrations. And we'd love to do this not just for nutrients and micronutrients, but also for the proteins, to see what the oceans are feeling, and what the microbes are doing, and what's controlling their growth. Next slide. And so this is an example of what we were trying to do, where we can see nitrogen stress closer to Hawaii, where that means this one microbe, Prochlorococcus, is low in nitrogen. To the north, where when you go south, they become low in iron as well, and there's a little bit of region where they're limited by both. And so that's an example of how we're using these proteins, but to go inside one particular microbe and see what's influencing their growth. Okay, next slide. I think I'm winding down. We could switch more to Q&A if you'd like at this point. This is sort of a schematic of what we're trying to do.
Coming from Mrs. Yagi's classroom, I think. I've seen a couple really good questions come in from them. Oh, it looks like they're in their middle of their class change right now.
Thank you.
But one of the questions that her students had are, why are there low oxygen zones or oxygen deficient zones around the Pacific? And like, are you seeing these in other areas of the ocean as well?
Yeah. So yes, there are low oxygen regions in the eastern Pacific, in the northern and southern hemispheres, and they sort of merge. Actually, they're so big that they merge, and as well as in the eastern Atlantic, as well as in the Indian Ocean. We see them, largely because of, we think, the physics of the region, as well as the high productivity of the biology there. But the reason for their expansion actually is one of the tricky things that we're not quite sure about. What's causing them to expand, and part of the thinking behind that is that there's warming of the upper water columns that is slowing down the circulation processes, that it's sort of shielding those waters from getting ventilated better. Great.
Coming in from the Lake and Park School in Seattle, we had questions. You shared with us some of your larger research equipment that you use to take water samples, but do you ever have scientists diving for water samples or diving to take any type of samples?
Yeah, we don't on this expedition, but I certainly know a lot of colleagues that dive. People who work on coral reefs and coastal ecosystems do a lot of scuba diving or snorkeling to get those samples. Out here, if we were to scuba dive or snorkel, we wouldn't see a whole lot, because the waters are amazingly blue out here. This is what we call blue ocean environments, where because there's not much land around and not much deep water nutrients coming up, the waters are incredibly clear, and that's why light can penetrate to almost 200 meters. But I also have a lot of colleagues that go down either in the human-occupied vehicles, like the Alvin. Woods Hole runs the Alvin submarine, and other nations have human-occupied vehicles as well. Or like the Falkor often has an autonomous vehicle on it to study the sediment ocean environment, or the hydrothermal vents at the bottom of the oceans. And so those are great tools to get down there and see what's happening in terms of where hydrothermal vents are and what kinds of biological and chemical and geological processes are happening at them. And that's really, it's really exciting. Those vehicles have a lot of capabilities now, both the autonomous ones and the human-occupied ones. Great. I think I have a few more slides. I can keep going in that, or I can take more questions.
Yeah, just a few, and then we have some more questions, but you can finish up these slides, and we'll go back to some of those questions.
Yeah, I don't mind. We can let's see. So yeah, this slide just sort of shows a schematic of what we're trying to do. Can we see the different ecosystems and how they're responding, and what's controlling, what micronutrients are controlling their growth, and what kind of reactions are happening. So the idea is that we're collecting water samples from the surface of the ocean all the way down. And this slide only goes to 150 meters or 200 meters, but really we're going down to 1,000 meters, and we're looking at what the whole ecosystem is doing and all the different microbes that we can measure effectively. And then I think the next slide is sort of a stepping back and what does this really mean? What is ocean deoxygenation potentially likely to do in the oceans? And just to recap, these are the purple areas are low oxygen regions of the oceans, and it's believed that they may be expanding. And one thing, what it means for the future is actually a really difficult thing to respond to. But one thing we do know is that these low oxygen regions are big sources of metals, in particular cobalt and iron, which are both required for growth by many microbes. And so if those regions start emitting more of those nutrients, it's likely to promote the growth of those microbes. And what does that mean for the whole ecosystem? Some of those microbes are really good at fixing nitrogen, so will that help bring more nitrogen in? Will it crowd out other microbes that don't need those nutrients quite as much? Will it change the cycling of iron and cobalt in the oceans? I think that is a clear yes at this point, even though we only have a limited amount of data. But it's that's why we're out here, to try and collect some of those answers. Great. So yeah, let's do more questions.
Yeah. So we have a few more questions that we've gotten from our online viewers, some of our online schools or students that are viewing online. And one question that a lot of students have is just, what is your typical day like? You talked a lot about how you need to split up your research and your sampling, but you can only do so much sampling at a time. So what does your typical day look like out on a research vessel?
Yeah, so out here we typically start with breakfast, and then I have a meeting with the captain and the crew to discuss the plans for the day up on the bridge. And then when we get to station, we begin science ops right away, and that will typically involve deploying what we call the CTV, which stands for the big rosette that has all of these bottles on it. Big sample bottles. It has 24 of these on a metal frame and all the sensors, and we'll deploy that first because that gives us a sense of what the water column is doing. And then that'll bring up a whole bunch of water, 24 bottles of water, and then we will usually immediately deploy the trace metal rosette, as we call it, which is another rosette that's designed to have as minimal amounts of metal as possible and doesn't use a metal wire, so that we avoid contaminating the samples. And then we'll switch to the pumps, which will pump. I think there are roughly about 1,000 liters per pump we've discovered on this cruise. So last night's cast did, I think over 6,000 liters of water were pumped underwater, and then all that biomass, all the microbes on the filters were brought up on deck. And then there's a lot of sample processing. That's a big part of it, and making measurements. There's also net tows and looking at the microbes under the microscope. And then that's all while we're living on the ship. Eating lunch and dinner. Sometimes when people want to kick back, we'll have a barbecue. We're gonna have a barbecue tonight, or sometimes we'll have a movie. But to be honest, we're working really hard a lot of the time. So a lot of it's organizing and trying to figure out what to do and where to put stuff and what experiments should be happening and coordinating.
That sounds like you guys are really busy during the entire cruise. And one thing, as you're conducting all this research, because we are running out of time here, one thing that a lot of students want to know is, even students that don't live in the Pacific or really close to the Pacific, what are some things that they can do? We talk about how these oxygen deficient zones are becoming larger, most likely due to human causes, global warming. So what are some things that students can do right now to help combat some of these effects and help protect not only the Pacific but their own local ecosystems and environments?
Yeah, that's a great question. I think one of the major things people can do is trying to reduce their carbon footprints, be it driving more fuel efficient cars and minimizing wasting fuel, leaving cars idling, and trying to bike more, and and trying to promote the idea of moving to a lower carbon economy through convincing other people and talking to your politicians. In addition, I think for the deoxygenation in particular, a lot of that's caused in coastal environments by too many nutrients coming out, and that means sometimes a lot of fertilizers that are being put on non-agricultural lands that aren't necessary, and so those are two big things, I think.
Great. I think that those are great suggestions, and there's a lot that students can do in their own communities that will have a large effect. So we wanted to say thank you so much for joining us today. I think this has been a really, really informative hangout, and you've taken us to a really cool place and showed us some things that people don't normally get to see. For those of you that want to follow along on the rest of their cruise, because they are pretty early into the cruise right now, you can go to schmidtocean.org. And there are links to that on our website. There you can follow along with daily blogs and videos. If you're watching the Hangout on our website, that link can be found in our classroom resources section. And we'll also be sharing some of that information on our social media. All of that is up on your screen right now. Stay connected with us through Facebook, Twitter, and Google+. And again, thank you so much, Miss Yagi's class, for joining us, Mac for talking to us, and for Schmidt Ocean Institute for helping to put this on. So thank you guys so much. Everyone have a great rest of your day.
Thank you. Thanks for everyone for participating.
Bye.